Hi everyone, this is Adil. Today I'm going to talk about layers of epidermis. There are five layers of epidermis starting from the stratum basil, which is the deepest layer of epidermis, and the stratum corneum, which is the uppermost layer of epidermis. Stratum basil is followed by stratum spinosum, then stratum granulosum, then stratum lucidum. Stratum basil is the layer which is developed first, and from the stratum basil there is a formation of stratum spinosum. Then stratum spinosum is followed by stratum granulosum, then stratum lucidum, then finally stratum corneum. In preterm babies or low birth weight babies, the stratum corneum is deficient because that is the last one to be formed. Now let's continue with the first layer to be formed, that is stratum basil. The stratum basil is a single cell layer having keratinocytes, and these keratinocytes are mitotically very active. Now there is a time which is known as epidermal turnover time or skin doubling time. It is that time which is taken by the keratinocytes to move from the stratum basil to the environment. As you can see here, this is the stratum basil and this is the stratum corneum, and this is the lowermost layer of stratum corneum. Keratinocytes take around 14 days to move from the stratum basil to the lowermost layer of stratum corneum, and further 14 days are taken by the keratinocytes to cross the stratum corneum. Now next 28 days are taken by the keratinocytes to be sloughed off into the environment. Let's calculate epidermal turnover time or skin doubling time. The first 14 days plus next 14 days plus these 28 days, that's equal to the 56 days. Now the epidermal turnover time is about 56 days. There is a condition which is known as psoriasis. In this condition, the epidermal turnover time is of only 4 days. It means that the movement of keratinocytes from the stratum basil to the environment is very rapid. Right. Now there's a controversy of epidermal turnover time. In different books or guides, the different values of epidermal turnover time are given. Like in Arvind Arora's guide, 28 days. In Nina Khanna's textbook, 60 days. In Manish Soni's book, 56 days. And in Rook's textbook, 52 to 75 days. I would like to suggest to you to remember this one, because anything which lies in this range is the correct answer. 56 days and 60 days both lies in this range. So both these are the right answers, and this one is the wrong answer, and don't remember this one. Now let's continue with second layer, that is stratum spinosum. Stratum spinosum is also known as prickle cell layer. Why this name is given? Because in between two keratinocytes there are some connections which are called spines or desmosomes. Now these desmosomes help in the attachment of two keratinocytes. This stratum spinosum is the thickest layer in the epidermis. Let's see this question. Desmosomes are prominently seen in which part of an epidermis? This question was asked in NEET PG 2018 and its right answer was stratum spinosum. Now let's see desmosomes in this diagram. Here is the stratum spinosum, this whole area, and these are the keratinocytes. In between the keratinocytes there are desmosomes, and these desmosomes help in the attachment of two keratinocytes, and these desmosomes are prominent in the stratum spinosum. These desmosomes are made up of desmosomal proteins, which are also known as calcium dependent adherent molecules, and these desmosomal proteins belong to gene family of cadherin, plaquin, and armadillo. And the examples of desmosomal proteins are desmoglin, desmocolin, desmoplakin, placoglobin, and placophilin. Any defect in these proteins lead to intraepidermal disorder. In intraepidermal disorder, the keratinocytes get separated. Whenever the keratinocytes get separated, the extracellular fluid accumulates and a blister is formed. Defect in desmoglin leads to intraepidermal disorder, which is known as pemphigus. Now let's continue with arrangement of desmosomal proteins. As you can see here, this is the keratinocyte, this is another keratinocyte. In between the keratinocytes there are desmosomes, and within the desmosome there are desmosomal proteins. Now as you can see here, this is the first desmosomal protein, that is desmoglin, and this is desmocolin. The desmoglin and desmocolin are attached to placoglobin and placophilin. Then placoglobin and placophilin is further attached to desmoplakin. Then desmoplakin is further attached to intermediate filaments. Intermediate filaments are also known as keratin filaments. Again, let's see the arrangement of desmosomal proteins in this diagram. This is the first keratinocyte and this is another keratinocyte. This black colored protein is desmoglin and this white colored protein is desmocolin. The desmoglin and desmocolin is attached to placophilin and placoglobin. Then placophilin and placoglobin is further attached to desmoplakin, and then desmoplakin is further attached to intermediate filaments. There are two junctions which are seen in electron microscopy. The first one is outer dense plaque and the second one is inner dense plaque. This outer dense plaque is formed by when desmoglin and desmocolin attach with placophilin and placoglobin, and the inner dense plaque is formed by when desmoplakin attaches with intermediate filaments. Hope you have enjoyed this video. Thanks.